recording. It's about to start. So that's working. And I'm going to share my screen. So start the broadcast. That should work as well. And the question is, can you see the screen? Does that work? The screen? Yes, we can. Yeah, that's great. That's a little bit like Bob the Builder, isn't it? Yes, we can. Fantastic. And Bernardo has kindly put up the link to Moodle again, so thank you very much. Nucleic acid is the death of, it's not, it's life. Oh, you've gone over the history of the discoveries. Have you done the history of nucleic acids? Not all of it. Okay. Just touched on it. Okay, well, bear with me, and maybe I can tell you a few nice stories about the whole thing. So let's make a start. And as I said, I will try to give it a little bit of a personal touch from my perspective. So this is really about how we found out about nucleic acids and what the developments were. And then in the session tomorrow, we will have a look a little bit into the future about the possibilities of nucleic acids, but also the dangers that are associated with it. And I really want you to get a sort of a feel for the people who did all the work that gave us these insights into our knowledge today. So, I mean, obviously, people usually start with Mendel, but also we mustn't forget that at that time when Mendel was alive, no, it's not in any MCQs or in any assessments. This afternoon, I just want to entertain you, right? And put things a little bit into context. So this, around the time of Mendel, people became interested in how are traits inherited. And the time was ripe for it. Darwin published his Origin of Species. Oh, by the way, did you know? No, I haven't told anyone. Did you know that I have a handwritten letter from Darwin, Charles Darwin's granddaughter, Angela Darwin? I received that from her as a thank you for looking after her while she attended an event at the university. I just thought I should mention that. So that's my link to fame. Anyway, Darwin did the evolution of species. He didn't know about Mendel. We are pretty sure about that. And Mendel, who is basically the grandfather of all the Mendelian genetics that you have discussed, he had a little bit of, well, he wasn't that famous. He published his the laws of inheritance as we know them in 1865, but not many people actually paid attention to it. And only about 35 years later, when Mendel has long gone, scientists really took note of these finds and sort of put one and one together and rediscovered Mendel's work, really. There's an interesting thing about Mendel's work, actually, which is related to statistics. Actually, the thing is, if you look at the original work by Mendel, the numbers look simply too good. And you would expect from somebody who's doing these experiments that for the first time, and not really knowing what they are doing, that there are larger uncertainties in them. So some people have questioned whether he did a little bit of creative data management, let's put it this way, but I don't know. Let's start with Mendel then. And the next person really that made a massive contribution was this guy, Friedrich Miescher. I don't know, have you discussed Miescher in any shape or form? No? Well, he was quite an interesting guy. He wanted to become a GP, but because he had a disability, he had problems with hearing. He was persuaded to go down more the route of a scientist in physiology. Oh, that's a bad one, Edith. People have been crucified for less than that. He was encouraged to do his sort of PhD on white blood cells. So he spent a lot of time on purifying the nuclei from white blood cells, actually. And he came across an interesting substance that he found quite annoying when doing his analysis of the nuclei, which was a sort of a gooey, viscous substance, which he called nuclein, because it was in the nuclei. And he found out, actually, that this stuff was made of predominantly sugar. Then there was compounds that contained nitrogen, and it was phosphate in it. And somehow it's rather strange, actually. He thought in later years that this nuclein might contain some kind of information. He once wrote that maybe this is some kind of alphabet in it. And he actually sort of came up with this idea. There might be some information in it. Oh, by the way, I've put up this slide presentation on Moodle under BI300, and there are also little recordings for each or most of the slides. So if you want a little bit more background information. Then, so we had this, you know, there is this nuclein, and also people were asking about inheritance. And then around 1900, 1902, these two gentlemen, Bovary and Sutton, independently thought about the experiments that Mendel did. There was a guy called De Vries in Holland, who also looked at that. If it's not on Moodle, well, then don't worry about the attendance. Let's just simply enjoy. And in order to explain this inheritance, people played with different models. Oh, thank you very much, Conrad. That's great. And Bovary and Sutton could explain certain inheritance traits if they broke down sort of this nuclein into chunks. And they did some interesting, or a little bit of microscopy with these things. And they managed to stain this nuclein. And that's where the notion of chromosomes is coming from. So they suggested that these chromosomes are actually the carriers of this bizarre Mendelian inheritance patterns. Another guy got interested in that, Phoebus Levens, and he decided to look at this stuff closer. He was originally from one of the Baltic states. I can't remember, Latvia or Lithuania. And he had to immigrate because that was used to be part of the Russian Empire. And there was a sort of a huge anti-Semitic propaganda at the time. So he and his family moved to the States, where he then studied medicine at the Massachusetts Institute of Technology. And he became also very interested in these nucleic acids that have been discovered. And he managed to analyze that stuff, and he found that it is actually made of deoxyribose. Then there are some nitrogenous bases and phosphate. So he did a proper analysis, and he came up with a structure for DNA for this compound. And he proposed that it is basically sort of a square where you have sugar, 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 phosphate, 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 and each of these sugars was then actually linked to ATGC. And that was and this repeated over and over and over again. So that was the Levin DNA structure. But he couldn't figure out actually how this actually, what the purpose of this thing actually could be, because there was no, it was just a repetition of ATGC. And so it wasn't quite clear how this thing could actually work. Another guy, and I'm pretty sure my colleague, Dr. Peter Ellis, mentioned him because he is a huge fan of Thomas Hunt Morgan. Did he mention Morgan in the 324? Yeah, rings a bell. Yeah. He actually, exactly. It's about the flies. And he actually then established that the inheritance is actually somehow linked to the chromosomes. And he, the family history for Morgan is quite interesting, because he grew up in the South, in the Confederate States of the United States. very wealthy family, but they fell on hard grounds after the Civil War. And so luckily, he then decided not to become a cotton field owner, but to go into science, luckily. And he fell totally in love with developmental biology. And he did a lot of work on how fertilized sea urchin eggs develop. And that was a fantastic model. He actually did not like fruit flies, but he still worked with them because the fruit flies have in their salivary glands very, very large chromosomes. I don't know whether you've seen them, these puffed up chromosomes, and they were fairly easy to look at. And so Morgan actually developed, in a way, sort of the first very crude map of genes. And so that was a really major breakthrough, showing that these chromosomes are actually responsible for inheritance. And he is considered really the grandfather or the grandfather of genetic mapping. Now, people got more and more interested in this strange substance, this DNA, and Frederick Griffith then did a really, really interesting experiment. And I think you have all seen this experiment, because I know that Peter Ellis puts that up, where he used the different strains of, what was it, a pneumococcus or a salmonella? No, it was salmonella, where he came up with this transforming principle. And you've seen this, this picture, this slide? You heard about the... So basically, he injected mice with the rough strain. Mice lived, no problem. He injected them with the smooth strain. Mice died. So that's this one here. He heat inactivated the smooth strain, injected it, and the mouse lived. And then he mixed the sort of the dead strain, the heat inactivated, with the harmless strain, and all of a sudden the mouse died, keeled over. And that was really bizarre, because you wouldn't expect that. And I think it really took them quite a long time to sort of believe in this experiment. There is something you do, and actually my suspicion is that this experiment here, this crucial experiment, actually happened either as part of a control or just simply by accident. Why would you want to mix these things? Do you want to just simply demonstrate that they, you know, it's all fine? And luckily, he got a really, really strange result. And like any good scientist, he decided, I'm not throwing that in the bin. This is something totally unexpected. Maybe he liked to kill mice, yeah. But then I wouldn't use that method. So he got interested and said, and thought, wait a moment, this is not right. It shouldn't happen. Let's dig a little bit deeper. So he identified a sort of a transforming principle. And this then was picked up later on in 1944 by Avery, MacLeod, and McCarthy, who repeated the experiment again, and they tried to figure out. They figured out that actually the DNA, deoxyribonucleic acid, is sort of the thing behind this transforming principle. And they did a lot of very clever and interesting experiments. And they then discovered that, yes, it must be the DNA. So that was in 1944. And then a few years later, Hershey and Chase actually used a very different approach, but they then could demonstrate that it really, really is DNA. It's DNA and not proteins that is the carrier of this transforming principle and also the carrier of hereditary information. And they used, in rather complex experiments, they used bacteria. So here that would be a bacterium. And they then they used blenders, and they had these phages, the sworn enemies of bacteria, and they then tried to separate them from each other, and they could show that the phages actually injected DNA into the bacteria and transformed these bacteria, but it was the DNA and not the proteins that was the carrier of the genetic information. And they did these smoothies, exactly. And 1952, that was really a very interesting year because people got really, really interested in this DNA stuff. And exactly, they knew it was the DNA because the empty shell of the phage, sort of the wrapper, stayed outside and they could remove it. They could remove the protein wrapper, i.e. the phage, but they had the DNA of the phage in the bacteria, and that changed the genetic information in the bacteria. And that was really the big thing. Now, at the same time, a lot of people started to wonder what does this DNA actually look like? And there were actually about 3 research groups to speak of who tried to figure out what is the structure of this strange DNA molecule. But before the DNA the structure could be identified, there was another piece of information which came from this chap, from Erwin Chargaff, who looked at the DNA, and he made a really bizarre observation. He found that when he looked at DNA, that the percentage of A's is pretty similar, almost identical, to the percentage of T's in the DNA. So percentage of A contained the same number of A's and T's, and it contained the same numbers of G's and C's. So immediately people thought, there's something weird going on. And then he made a big mistake which made him doubt everything, really, because he then looked at RNA. And of course, with RNA, this what has been known as Chargaff's rule, that it's the same number of A's and T's and G's and C's. For RNA, it doesn't apply. And he couldn't explain that. He got really lost because, what's so different about these two things? They are both nucleic acids. They have A's. Okay, he figured out that there is no T in RNA. There's a U in RNA, but that doesn't matter really. But this Chargaff rules did not apply to RNA, and it totally threw him off the scent. Nevertheless, Chargaff's rule for DNA was out. And then these three people absolutely, yeah, it's an interesting thing. And then there were three big groups working on trying to figure out what this DNA might look like. There was the group around Maurice Wilkins in King's in London, and the group of Henri Bragg in Cambridge, and there was a fourth group, and they were all absolutely scared shitless of this. Sorry, a third group. They were absolutely scared shitless of this third group. There was the group around Linus Pauling in the US. And everybody was worried about Linus Pauling because he had lots and lots of money, and he was an incredibly smart person. So there was sort of a race to find out what might be the structure of this weird DNA molecule. Now, there is a very, very interesting story about the relationship between these British groups. There is an absolutely fantastic short movie about the story of betrayal. And I would really recommend you watch that, because it's all about, yes? James Watson is still alive. And I tell you a story about him in a minute. The whole story is really nicely summarized in this movie, or in this documentary. It was really this person here, Rosalind Franklin, who did basically all she did a lot of work for the whole thing, and she was miles ahead of everybody else. She was an incredibly gifted crystallographer who was originally, she's English, was English, but she got a job in Paris, and then was where she really learned X-ray crystallography, and she really refined her skills. She was the expert in the world for crystallography. And she then got a position at King's College. And the lab manager, actually, this guy, Maurice Wilkins, was on holiday when she arrived. And the overall chair of the institute then said, okay, Rosalind, I want you to start working on DNA structures. And Wilkins was also already working on the structures, but because he was on holiday, Rosalind Franklin said, well, okay, good, let's set up the lab. And maybe, and she started the work. Wilkins came back from his holiday and found that Rosalind Franklin has taken over the lab, has massively improved it, and also worked with Maurice Wilkins, PhD student. And they went from discovery to discovery. Wilkins was absolutely pissed off. He hated her with a passion, and there were very different personalities as well. It was a total personality clash. Rosalind Franklin was outgoing, was outspoken. She did not take any prisoners, whereas Maurice Wilkins was a very shy person. And it was a totally toxic relationship. And it was fostered really by the head of the institute not having clearly communicated what he expected from people. So that was a sort of a toxic constellation at King's College, and they worked really against each other. And then there was this group in Cambridge where an American guy called James Watson joined this group around Francis Crick, and they were both PhD students. And they decided that they want to use model building as a sort of, how shall I say, as an entrance into finding the structure of DNA. They knew Chargaff's rules, they knew it was deoxyribose, they knew sugars, and so they played around with models. And however, they needed data. Now, it turns out that Wilkins was a good mate of Francis Crick's. And and because Wilkins hated Rosalind Franklin, he sort of leaked information to the Cambridge group. And there was a fierce competition between them. James Watson actually approached Rosalind Franklin and said, shall we work together? We really need your data. And she said to him, well, yes, this is exactly what she said. Because she thought they know nothing. Watson was absolutely outraged that she did not want to share her unpublished results with him. And at one point, this famous picture, photograph 51, that Rosalind Franklin and her PhD student, Raymond Gosling, has taken through X-ray crystallography. Wilkins saw this picture, which was absolutely Rosalind's property. And somehow, nobody knows exactly how, it found its way into James Watson's hands. And it is reported that his jaw dropped when he saw this structure. This is actually the B conformation of DNA, which is more difficult to analyze. People used the A conformation so far. And Watson and Crick based their models on the A conformation, but they didn't get anywhere. But when Watson saw this picture, everything made sense. Everything made sense because this cross here of the diffraction pattern shows that it is a helix, and actually it is a double helix. So first he invited her, and first the Cambridge group invited the King's group before they saw this picture. And Rosalind really made fun of the clumsy model that they had developed. Watson said to her she doesn't know anything about X-ray crystallography, which of course incensed her even more. However, they then came up, once they had this picture, everything fell into place. So here is what they interpreted from this picture. The cross means the helix, the diamonds mean that the phosphate sugar backbone is on the outside. And they could even calculate the parameters. And Watson and Crick did their addition. They just modeled it and found that there is a minor and major groove. And knowing Chargaff's rules, this was the Chargaff's rule, they put 2 and 2 together and they came up with the double helix. And this was how they actually built the model. And Rosalind, and then they showed it to Franklin and Gosling, and immediately they realized that this was correct. But they couldn't publish the paper because it was based on basically theft and deception. So what they did instead was they decided to approach the publishers of the journal Nature and struck a deal where Watson and Crick published their model first. Then there was another article, I can't remember, that was by Maurice Wilkins and a co-worker, the second paper, and the third paper, where all the work was done by Franklin and Gosling. That was the third paper. And Nature was sort of influenced by the Cambridge group, and they sort of edited the Franklin paper in such a way that it was written as a confirmation of the ideas of Watson and Crick. So in a way, it was totally the other way around. Watson then in 1962 or 1963 also published a book where he, a really vile book about his relationship, how they found the double helix. He took all the credit, and he really ridiculed Rosalind Franklin in this book. It was written so badly, actually, that Harvard Press refused to publish it. But James Watson did not appear as a very nice person then. I have to say, I met him. I met James Watson at a presentation, at a talk, a conference, I think it was in 1986, when he tried to get money together for the Human Genome Project. Yes, I attended the conference, but I didn't speak to him. Everybody was sort of kissing his backside because he was the big man, but actually, he made a few remarks in this talk where I thought, really? I don't think so. And I think you are a racist but never mind. I better get rid of this because it's not nice to say that about people. So anyway, the rest is history. In 1962, they got the Nobel Prize, and unfortunately, Rosalind Franklin died in 58, most likely because, well, she died of cancer, which she probably got because she worked with a lot of X-rays during her time. So in a way, she died because of her work. So, however, when they discovered this structure, the famous double helix, Watson and Crick, to their credit, immediately realized that this uh, double helix actually lends itself to this semi-conservative replication, which you have done through, and you have heard about the famous Mendelssohn-Stahl experiment, where they looked at how DNA can actually replicate. And this double helix structure really lent itself to this semi-conservative replication model. So really everything is falling into place. And that's the nice thing about it. Now, of course, the story carries on because then people started to really work with that stuff. And in 1972, again, people carried on, and the groups around Boyer, Berg, and Cohen invented what we call today recombinant DNA technology. Berg and his group actually discovered that there are enzymes that can specifically cleave certain DNA sequences. He found what we call nowadays the restriction enzymes. Restriction enzymes. So these are enzymes that cut DNA at particular sequences. And Boyer and Cohen found that there are sort of inherited elements in bacteria that are circular and pretty small. You know what they are called in bacteria? What are these circular elements? They are the plasmids, absolutely. So they are the plasmids. And now you need to put things together. We have restriction enzymes that can cut DNA, and we've got plasmids as sort of vehicles. Now you can combine these things and put them together and create what is called recombinant DNA. And this actually led to an enormous boost of technology. But of course, you needed a way to verify that what you have produced when you did these recombinant DNA stuff, that you actually got the right things. Now, I probably should go back to this one here. These restriction enzymes are usually found in bacteria. And It was sort of at the beginning of this recombinant DNA revolution, if you like. They had to be, these restriction enzymes had to be very laboriously purified from different bacterial strains. And I remember when I did my master's in genetics, that must have been 85, 86, there were only a handful of these restriction enzymes around that people could use to do recombinant DNA. And our Bible, I remember our Bible was the New England biology catalog, because that was the leading company at the time in 1985 that that produces these restriction enzymes. And I remember in the T-bar, it was almost like the Pirelli catalog. People were really eager to get the latest edition of the NEB catalog, because they could look whether there is a new enzyme has been purified, and you could buy it, so that your particular sequence that you are working with or that you had in your DNA that you could cut. So it was really, really exciting when this catalog came out, and the list of restriction enzymes that are now available is, you know, a mile long. So it's no longer a big problem to cut DNA at whatever sequence you want. But of course, you had to make sure that you can actually read the DNA. You know what the positions of the bases were. And this is where this guy, Frederick Sanger, one of the big heroes in molecular biology, comes into play. He was an absolutely remarkable person. He won the Nobel Prize twice. One of the very few people who got this honor twice. One for elucidating the amino acid sequence of a protein, and the second Nobel Prize for what is now called the Sanger sequencing method. And we will talk about the Sanger sequencing, how this actually works. I will show you in the lecture tomorrow the principle of this. But with this method, it became possible to sequence, to get the base, the order of the bases of a DNA fragment without a lot of time and effort. And the original version, how you did this sequencing, was not with these swanky colors that you see here. It was done with radioactively labeled nucleotides that you could use. And I remember I used this method, this Sanger sequencing, with radioactive labeled nucleotides. It was pretty dire, the whole process, because you had to use a lot of radioactivity, especially phosphorus, which is a very heavy sort of beta emitter. So you got quite a decent exposure, where if you were not careful, when you worked with these substances. And I remember when I did my master's in 85, that the first sequencing attempt was unsuccessful. In fact, this went over my trousers, and I think I told you that, that I had to leave then my genes in the hot lab and go home via public transport without trousers. But luckily it was winter and I had a long coat, so nobody knew that I was basically without trousers on the bus. No, you know, people do wear underpants, so not to worry. So Sanger actually revolutionized the way how we can sequence DNA, how we can find the DNA sequence of whatever fragment that we are looking at. And it was at this time when, again, the people started to produce actually the first recombinant proteins. And in 1978, the American company Eli Lilly got filed for a patent for the production of recombinant human insulin produced in bacteria and verified by the Sanger sequencing method. One of the big breakthroughs. And people then started thinking about, can we actually do the human genome with that? In 1983, another big person entered the scene, although he didn't know it that he would be so big. Carrie Mullis. He seemed to be a very interesting character because he was an academic, then left academia, wrote fiction, and then went into a company. He was considered a totally loose cannon. He was wild, but bloody hell, he was good because he came up with the concept. He made sort of the concept of PCR viable, that you have two primers flanking the DNA bit of interest, and just simply by changing the temperature, you can run these PCR cycles. PCR was invented in 1983, and as I said, when I did my master's in 85, we were thinking of actually trying to do a PCR reaction. But of course, we didn't have a PCR machine or something like that. That's absolutely right. It wasn't, PCR didn't exist then. But and we thought, let's do it. Let's try to figure out how we can do it. So we thought, okay, we asked a PhD student, and we had 3 different water baths at that time, where the student had to dip the samples in. But one of the problems was we were quite curious whether it would work, but we didn't have the right primers and it never worked. But the company that Mullis was working in, they then actually used and refined the method. He got a bonus of $10,000 US, and the company sold then the patent for, I think, 30 million. And he was absolutely pissed off with that. Well, and over the last few decades, things have developed further. We are now at a point where basically everybody does PCR. But the story doesn't end here, because this year the Nobel Prize went to Jennifer Doudna and Emmanuel Charpentier for their discovery of sort of the universal restriction enzyme CRISPR-Cas9. And should you decide to stay on after your degree and do a master's, you will do experiments, incredibly interesting experiments with CRISPR-Cas9, which allows people to edit and modify whole genomes. We are not yet at a point where we can use that to cure people from genetic diseases, although there are rumors that some groups have actually tried it. At the moment, this CRISPR-Cas9 system makes too many errors, but this is where gene editing comes in, and this is the future of genetics. So let's stop here, and tomorrow let's have a look at the Sanger sequencing, what was so revolutionary about it, how this has developed. And I should probably say I left out a step here, because in 1990, on the 1st of October 1990, the Human Genome Project started. This was probably the only project, massive project in science that went under budget and finished on time, because it finished around 2003. It was declared that the human genome had been sequenced with the Sanger method, and with the method using also a lot of PCR. So without Sanger and Mullis, the Human Genome Project would not have been possible. And now we are there where we make use of this information, and how we make use of all this information, I'm going to tell you tomorrow. So stay tuned, and I'm looking forward to seeing you tomorrow to the future of DNA. Thank you for watching, and I hope you found this interesting. See you tomorrow.